Hello again. Okay, in the next this next video, this follows the video on the light-dependent reactions, and this is the light-independent reactions, or sometimes referred to as the Calvin cycle, named after Melvin Calvin, who first did all of the biochemistry to measure all of this. So it's during the Calvin cycle, the light-independent reactions, when carbon dioxide is fixed. So carbon dioxide is inorganic carbon in the atmosphere. It needs to be captured, so that carbon and the oxygen need to be captured within biological molecules. So if you remember back to the previous video, I said one of the important features of the light-dependent reaction is it's capturing all of the hydrogen that makes up biological molecules. Well, all of the carbon and all of the oxygen comes from carbon dioxide in the atmosphere from the light-independent reactions. Okay, so we begin by thinking about, firstly, how that carbon dioxide gets in. So it's in the atmosphere, it then diffuses down into the leaf via the stomata, through the spaces in the spongy mesophyll until it reaches one of the mesophyll cells, tends to be, or perhaps most often the palisade mesophyll cells. It dissolves on the water on the wall of the palisade mesophyll and then diffuses through the membrane into the cytoplasm. From the cytoplasm, it diffuses through the two membranes of the chloroplast and into the stroma. So the light-independent reactions are taking place in the stroma. These reactions are catalyzed by enzymes, and therefore the light-independent reactions are temperature sensitive. Okay, an increase in temperature may mean that those enzymes are forming enzyme substrate complexes faster, but a temperature that's too great may cause those enzymes to denature. We'll come back to that. I'll do a follow-up video about being a really good vegetable grower later on. Okay, so carbon dioxide is diffused eventually all the way into the stroma. In the stroma, the one carbon in carbon dioxide combines with a molecule that contains 5 carbons. Now this molecule is called ribulose bisphosphate, or ribulose 1,5 bisphosphate, but you can refer to it as RU, lowercase u, capital R, BP. So ribulose bisphosphate. It has 5 carbons, and it combines with oxygen in this enzyme, Rubisco. Ribulose bisphosphate carboxylase oxygenase. Just call it Rubisco, it's fine. Okay, so Rubisco is the most abundant enzyme on Earth. It's everywhere, well, in all green plants at least. So carbon dioxide is able to bind to the Rubisco with the RuBP. The carbon dioxide combines with the two, and then for a very short time that produces a 6 carbon molecule, but it's so unstable that we can ignore that. And from that 6 carbon molecule, we get 2 molecules of 3 carbon molecules two of those, called glycerate 3 phosphate. Now glycerate 3 phosphate can just be abbreviated to GP. Okay. Or sometimes you may see it abbreviated to G3P. are a load of other terms that you may see on the internet. But if we can just stick to glycerate 3-phosphate or GP, that's absolutely fine. Okay, in the next stage, this is where the products of the light-dependent reaction come into play. Sorry, I'm just going to go back a step. We'll call this first stage carbon fixation. So fixing carbon into a biological molecule, so we can consider glycerate 3-phosphate to be a biological molecule, fixing it from the atmosphere. So it's no longer inorganic, it's now an organic molecule. Okay. Glycerate 3-phosphate then undergoes a reduction. So it is reduced. Reduction is the gain of electrons and the gain of protons. So where does it get those electrons and protons from? Well, from the products of the light-dependent reaction. So you remember the light-dependent reaction produces reduced NADP. So reduced NADP donates electrons and hydrogen to the glycerate 3-phosphate to produce 2 3-carbon, I'll draw that there, triose phosphates, or can just be abbreviated to TP. So as the reduced NADP donates it, it becomes oxidized. So we've now got that oxidized the oxidized NADP, which can then go back to the light-dependent reactions that are taking place at the thylakoid, collecting more hydrogen and more electrons. Now, this second stage is reduction. Reduction is endergonic, or similar to endothermic. It requires energy. So where does that energy come from? Probably guess. The other product of the light-dependent reactions was ATP. So that ATP from the light-dependent reactions is used or provides energy through hydrolysis. So the ATP is hydrolyzed down into, I'm sorry about the squeak, ADP and inorganic phosphate, releasing energy. Okay, so for the reduction of GP into TP, that requires the reducing power. It requires the hydrogen and electrons from reduced NADP and energy from the hydrolysis of ATP. So we'll call this second stage reduction of GP to TP. Okay, now in the third stage, and this is shown on page 20, and the diagram I suppose follows on page 21. Triose phosphate is used to regenerate RuBP. Okay. Now this requires more energy. That energy comes from the hydrolysis of ATP into ADP plus inorganic phosphate. And you're probably wondering at this point, well, two things. What is the point of all of that, if we've just come back to the start? And how did 2 3-carbon triose produce 1 5-carbon ribulose bisphosphate? Well, the answer to that comes from the idea that this reaction is taking place sort of continually, and we get an accumulation of triose phosphate. In your notes on page 22, there is a balanced diagram. Okay, that balanced diagram helps some people understand this process. Other people might just think, well, I don't care about the balanced diagram. You don't need to know it specifically for the exam, so you can just skip through this bit. But it is fairly important, I suppose, because we've not actually mentioned, how we get that glucose that is the product of photosynthesis. Okay, so this third stage, I've not labelled that yet, is it? Is called the regeneration of RuBP, ribulose bisphosphate, requiring energy from the hydrolysis of ATP. Okay, let's imagine then that the cycle turns 3 times. Okay, meaning we've got 3 carbons going in, 3 ribulose bisphosphates, producing 6 glycerate 3-phosphate. Those 6 glycerate 3-phosphate would then be reduced to 6 triose phosphate. Each time it turns 3 times the Calvin cycle, 1 molecule of triose phosphate can be taken away. So, after 3 turns of the Calvin cycle, that produces 1 triose phosphate, 6 triose phosphate, 1 of which can go off. Okay, and this triose phosphate can then be used to make organic biological molecules. We'll come back to exactly how that is in a moment. Okay, so turns 3 times. We had 6 triose phosphates. Now we're down to 5 triose phosphates. So we know triose phosphate contains 3 carbons. So 5 times 3 means we have 15 carbons. Okay, it's turned 3 times. There are 6 of them there. Let's just change that to 6. We'll change that one to 6 as well. One of the triose phosphates has disappeared, meaning we've now got 5 triose phosphates that are going back here. 5 times 3 is 15, or 3 times 5. So 5 triose phosphates are used for the regeneration of 3 RuBPs. Okay, leaving 1 triose phosphate to be turned into organic molecules, into biological molecules. Right, let's go one step further and we'll look at how the diagram on yours is balanced. Let's imagine that this turns 6 times. So the Calvin cycle turns 6 times. That means we're going to have, 6 carbon dioxide. Think about the overall equation for photosynthesis, 6 carbon dioxide in. That's going to produce 12 glycerate 3-phosphates, GPs. Those 12 GPs are going to be reduced to 12 triose phosphate. This, like I said, with each 6 that are produced, 1 can be taken away. Now, we've got 2 triose phosphate, 2 3-carbon triose phosphate can be taken away from the Calvin cycle and used to make biological molecules. 3 plus 3, for example, makes a hexose. We could make, with 2 of those, a 6-carbon hexose sugar. For example, glucose. So there's the magic of it. I get excited talking about this. Okay, each time this turns around 6 times, we can remove 2 triose-phosphates. Those 2 triose-phosphates can then be used to make 1 glucose molecule, a hexose sugar. So again, if we go back to that overall equation for photosynthesis, 6 carbons in, or 6 carbon dioxides in, and we get that 1 glucose molecule being produced down there. That will leave us with 10 triose-phosphate, which go back to make 6 ribulose bisphosphates back here. Okay, so that's pretty amazing, I think. Let's think about all of the atoms that have come together to make our first biological molecule. All of the carbon and all of the oxygen in all biological molecules comes from the atmosphere. And it is fixed in this process, in the light-independent reactions, or the Calvin cycle. It combines with hydrogen from NADPH, the reduced NADP. Where did that hydrogen come from? Well, photolysis in the light-dependent reactions. So what we're saying here is we've created the first biological molecule. But it's not just glucose that can be produced here. Sort of running out of space a little bit. Okay. 2 triose-phosphates can make 1 molecule of glucose. Triose-phosphate can also be used if triose-phosphate and glycerate 3-phosphate are combined together. So if we have a triose-phosphate plus a glycerate 3-phosphate, well, the glycerate 3-phosphate can produce fatty acids. The triose-phosphate, 3 carbons, very similar to glycerol, that can be used to make glycerol. Combine those 2 things together, and we have lipids, triglycerides. Okay, so the triose-phosphate and the glycerate 3-phosphate from the Calvin cycle has now made triglycerides, or lipids. Amazing. So, okay, we've made carbohydrates and we've made lipids. I'm really running out of space now. Right, if you take one of those triose-phosphates and some nitrates that have been absorbed from the soil, and we'll cover this a bit more when we do the nitrogen cycle. So, NO3, 2 minus, plus some of the triose-phosphate, we can make amino acids. Or, with a little bit of different biochemistry, you can make nucleic acids. Okay, both containing nitrogen. Obviously, nucleic acid would have to take some phosphorus from the phosphorus cycle. I think you probably gathered though, for a start you might have gathered this excites me. But secondly, when I say that all biological molecules are the product of photosynthesis, this is why. Okay? It's captured carbon from the atmosphere, oxygen from the atmosphere, hydrogen from water. Combine those things together, carbon, hydrogen and oxygen, you can make carbohydrates, glucose for example, or lipids, triglycerides. Combine that with some nitrates that have been absorbed from the soil, and it's possible to make amino acids and nucleic acids, nitrates and phosphates from the soil, to make nucleic acids. 4, 3, minus is needed as well to make those nucleic acids. So photosynthesis isn't just important for making glucose, it's important for producing all biological molecules. So again, going back to essays, if you want a really good standout comment on the importance of this process of photosynthesis, well, it's captured all of the carbon, all of the oxygen, all of the hydrogen. It may combine it with nitrates and phosphates to produce all biological molecules. Plants do that. Plants are referred to as producers. They have produced those biological molecules. Plants can then be consumed by animals, and in consuming them, all an animal is doing is taking those products of photosynthesis and assimilating it, making it into their own biological molecules, cells, tissues, organs, etc. So when you come to do your essay, if you want to talk about importance, there's not much in biology that is more important than this. Okay, I hope that was helpful. See you later.